Hello students, I welcome you all. Today I am going to deliver a lecture on one of the basic topics of cell biology, that is cell wall and extracellular matrix. Let's start with the objectives of the lecture. Number first, to acquaint the students about the structure of cell wall both in prokaryotes as well as in eukaryotes. Number second, to understand the extracellular matrix. Now let's talk about the introduction of lecture. Although cell boundaries are defined by the plasma membrane, many cells are surrounded by an insoluble array of secreted macromolecules. Cells of bacteria, fungi, algae and higher plants are surrounded by rigid cell walls which are an integral part of the cell. Although animal cells are not surrounded by cell walls, most of the cells in animal tissues are embedded in an extracellular matrix consisting of secreting proteins and polysaccharides. The extracellular matrix not only provides structural support to cells and tissues but also plays important roles in regulating cell behavior. Interactions of cells with the extracellular matrix anchor the cytoskeleton and regulate cell shape and movement. Likewise, direct interactions between cells are key to the organization of cells in the tissues of both plants and animals, as well as providing channels through which cells can communicate with their neighbors. Now, what is cell wall? The rigid cell walls that surround bacteria and many types of eukaryotic cells like fungi, algae and higher plants determine cell shape and prevent cells from swelling and bursting as a result of osmotic pressure. Despite their common functions, the cell walls of bacteria and eukaryotes are structurally very different. Bacterial cell walls consist of polysaccharides cross-linked by short peptides which form a covalent shell around the entire cell. In contrast, the cell walls of eukaryotes are composed principally of polysaccharides embedded in a gel-like matrix. Rather than being fixed structures, plant cell walls can be modified both during development of the plant and in response to signals from the environment. So, the cell walls of plants play critical roles in determining the organization of plant tissues and the structure of entire plants. Now, what are bacterial cell walls? The rigid cell walls of the bacteria determine the characteristic shapes of different kinds of bacterial cells. For example, some bacteria such as E. coli are rod-shaped, while as others are spherical. For example, Pneumococcus and Staphylococcus are spiral-shaped. For example, the spirochete Trypanosoma pallidum, which causes syphilis. In addition, the structure of the cell walls divides bacteria into two broad classes that can be distinguished by a staining procedure known as Gram stain, developed by Christian Gram in 1884. The bacterial cell wall consists of peptidoglycan or murine, a polymer consisting of N-acetyl glucosamine and N-acetyl muramic acid, and short chain of amino acids. Alternating N-acetyl muramic acid and N-acetyl glucosamine molecules form a carbohydrate backbone, the glycan portion. Rows of N-acetyl glucosamine and N-acetyl muramic acid are linked by polypeptides, that is the peptide portion. The structure of polypeptide cross bridges may vary, but they always have a tetrapeptide side chain which consists of 4 amino acids attached to N-acetyl muramic acids. The amino acids occur in alternating D and L forms. While peptidoglycan is present in almost bacterial cell walls, there are two basic variations of structure seen in most bacterial cells, one described as gram positive and the one described as gram negative. Now, the gram positive cell wall. Gram positive cell wall consists of many layers of peptidoglycan and also contain teichoic acid. Teichoic acid may, number first, bind and regulate movement of cations into and out of the cell. Next, prevent extensive wall breakdown and possible cell lysis during cell growth. And number third, it provides much of the cell wall's antigenicity. Now let's talk of gram-negative bacterial cell walls. Gram-negative bacteria such as E. coli have a dual membrane system in which the plasma membrane is surrounded by a permeable outer membrane. These bacteria have thin walls located between their inner and outer membranes. In contrast, gram-positive bacteria, such as common human pathogen Staphylococcus aureus, have only a single plasma membrane, which is surrounded by a much thicker cell wall. Despite these structural differences, the principal component of cell walls of both gram-positive and gram-negative bacteria is a peptidoglycan, which consists of linear polysaccharide chains cross-linked by short peptides. Because of this cross-linked structure, the peptidoglycan forms a strong covalent shell around the entire bacterial cell. Interestingly, the unique structure of their cell walls also makes bacteria vulnerable to some antibiotics. Penicillin, for example, inhibits the enzyme responsible for forming cross-links between different strands of the peptidoglycan, thereby interfering with cell wall synthesis and blocking bacterial growth. Now, the eukaryotic cell walls. In contrast to bacteria, the cell walls of eukaryotes, including fungi, algae, and hair plants, are composed principally of polysaccharides. The basic structural polysaccharide of fungal cell wall is chitin, which also forms the shells of crabs and the exoskeletons of insects and other arthropods. Chitin is a linear polymer of N-acetylglucosamine residues. The cell walls of most algae and hair plants are composed principally of cellulose, which is the most abundant polymer on Earth. Cellulose is a linear polymer of glucose residues, often containing more than 10,000 glucose monomers. The sugar residues in both chitin and cellulose are joined by a 1,4 linkage, which allow the polysaccharides to form long straight chains. Following transport across the plasma membrane to the extracellular space, single polysaccharide chains associate in parallel with one another to form microfibrils. Cellulose microfibrils can extend for many micrometers in length. Within the plant cell wall, cellulose microfibrils are embedded in a matrix consisting of proteins and two other types of polysaccharides, one hemicellulose and second pectins. Hemicelluloses are highly branched polysaccharides that are hydrogen bonded to the surface of cellulose microfibrils. This stabilizes the cellulose microfibrils into a tough fiber, which is responsible for the mechanical strength of plant cell walls. The cellulose microfibrils are cross-linked by pectins, which are branched polysaccharides containing a large number of negatively charged galacturonic acid residues. Because of these multiple negative charges, pectin binds positively charged ions such as calcium 2 positive and trap water molecules to form gels. An illustration of their gel-forming properties is seen in jams and jellies that are produced by the addition of pectins to fruit juice. In the cell wall, the pectins form gel-like network that is interlocked with the cross-linked cellulose microfibrils. In addition, cell walls contain a variety of glycoproteins that are incorporated into the matrix and are thought to provide further structural support. Algae typically possess walls constructed of glycoproteins and polysaccharides. However, certain algal species may have a cell wall composed of silicic acid. Alginic acid is a common polysaccharide in the cell walls of brown algae. Monocyl form microfibrils in the cell walls of a number of marine green algae such as Stabularia, as well as in the walls of some red algae like Bangia and Porphyra. Sulfonated polysaccharides occur in the cell walls of most algae. Those common in red algae include agarose, carrageenan, porphyrin, fusarilan, and phenoran. The group of algae known as the diatoms synthesize their cell walls from silicic acid. Both the structure and function of cell walls change as plant cells develop. The walls of growing plant cells, called primary cell walls, are relatively thin and flexible, allowing the cells to expand in size. Once cells have ceased growth, they frequently lay down secondary walls between the plasma membrane and the primary cell wall. Such secondary cell walls, which are both thicker and more rigid than primary walls, are particularly important in cell types responsible for conducting water and providing mechanical strength to the plant. Primary and secondary cell walls differ in composition as well as in thickness. Primary cell walls contain approximately equal amounts of cellulose, hemicellulose, and pectins. In contrast, the more rigid secondary walls generally lack pectin and contain 50 to 80% cellulose. Many secondary walls are further strengthened by lignin, a complex polymer of phenolic residues that is responsible for much of the strength and density of wood. The cellulose fibers of primary walls appear to be randomly arranged, whereas those of secondary walls are highly ordered. Secondary walls are frequently laid down in layers in which the cellulose fibers differ in orientation, forming a laminated structure that greatly increases cell wall strength. One of the critical functions of plant cell walls is to prevent cell swelling as a result of osmotic pressure. In contrast to animal cells, the plant cells do not maintain an osmotic balance between their cytosol and extracellular fluids. Consequently, osmotic pressure continually drives the flow of water into the cell. This water influx is tolerated by plant cells because their rigid cell walls prevent swelling and bursting. Instead, an internal hydrostatic pressure called turgor pressure builds up within the cell, eventually equalizes the osmotic pressure and preventing the further influx of water. Turgor pressure is responsible for much of the rigidity of plant tissues, as is readily apparent from examination of a dehydrated wilted plant. In addition, turgor pressure provides the basis for a form of cell growth that is unique to plants. In particular, plant cells frequently expand by taking up water without synthesizing new cytoplasmic components. Cell expansion by this mechanism is signaled by plant hormones, that's auxins, that activate proteins called expansions. The expansions act to weaken a region of the cell wall, allowing turgor pressure to drive the expansion of the cell in that direction. As this occurs, the water that flows into the cell accumulates within a large central vacuole, so the cell walls expand without increasing the volume of its cytosol. Such expansion can result in a 10 to 100-fold increase in the size of plant cells during development. As cells expand, new components of the cell wall are deposited outside the plasma membrane. While the matrix components, including hemicelluloses and pectins, are synthesizing in the Golgi apparatus and secreted there, cellulose is synthesized by the plasma membrane enzyme complex, that is cellulose synthase. Cellulose synthase is a transmembrane enzyme that synthesizes cellulose from UDP glucose in the cytosol. The growing cellulose chain remains bound to the enzyme as it is synthesized and is translocated across the plasma membrane to the exterior of the cell through a pore created by multiple enzyme subunits. A similar mechanism is used to synthesize chitin and hyaluronan, a component of the extracellular matrix. In expanding cells, the newly synthesized cellulose microfibrils are deposited at right angles to the direction of the cell elongation, an orientation that is thought to play an important role in determining the direction of further cell expansion. Interestingly, the cellulose microfibrils in elongating cell walls are laid down in parallel to the cortical microtubules underlying the plasma membrane. These microtubules appear to define the orientation of newly synthesized cellulose microfibrils, possibly by determining the direction of movement of the cellulose synthase complex in the membrane. The cortical microtubules thus define the direction of cell wall growth, which in turn determines the direction of cell expansion and ultimately the shape of the entire plant. Cell walls from different plant tissues such as leaves, stems, roots, and flowers consist primarily of cellulose but differ in their matrix components and perhaps in the organization of their cellulose fibrils. There are at least 10 different cellulose synthase enzymes, and each plasma membrane enzyme complex contains 2 or more different forms, perhaps accounting for the varying organization of cell wall in different tissues. Now, the extracellular matrix. The extracellular matrix, commonly known as ECM, is a non-cellular component present within all tissues and organs and provides not only essential physical scaffolding for the cellular constituents, but also initiates crucial biochemical and biomechanical cues that are required for tissue morphogenesis, differentiation, and homeostasis. The importance of extracellular matrix is vividly illustrated by a wide range of syndromes which can be anything from minor to severe that arise from genetic abnormalities in ACM proteins. Although fundamentally the ACM is composed of water, proteins, and polysaccharides, each tissue has an ECM with a unique composition and topology that is generated during tissue development through a dynamic and reciprocal biochemical and biophysical dialogue between the various cellular components, for example, epithelial, fibroblast, adipocyte, endothelial elements, and the evolving cellular and protein microenvironment. Indeed, the physical topology and biochemical composition of the ECM is not only tissue-specific but is also markedly heterogeneous. Cell adhesion to the ECM is mediated by ECM receptors such as integrinase, discordant domain receptors, and syndicans. Adhesion mediates cytoskeleton coupling to the ECM and is involved in cell migration through the ECM. Moreover, the ECM is a highly dynamic structure that is constantly being remodeled either enzymatically or non-enzymatically, and its molecular components are subjected to a myriad of post-translational modifications. Through these physical and biochemical characteristics, the ECM generates the biochemical and mechanical properties of each organ, such as its tensile and compressive strength and elasticity, and also mediates protection by a buffering action that maintains extracellular homeostasis and water retention. In addition, the ECM directs essential morphological organization and physiological function by binding growth factors and interacting with cell surface receptors to elicit signal transduction and regulate gene transcription. The biochemical and biomechanical protective and organizational properties of the ECM in a given tissue can vary tremendously from one tissue to another. For example, lungs versus skin versus bone, and even within one tissue, for example, renal cortex versus renal medulla, as well as from one physiological state to another, for example, normal versus the cancerous. The ECM is composed of two main classes of macromolecules, protein, proteoglycans, and fibrous proteins. The extracellular matrix includes the interstitial matrix and the basement membrane. Interstitial matrix is present between various small cells, for example, in intracellular spaces. Gels of polysaccharides and fibrous proteins fill the interstitial space and act as compression buffer against stress placed on the ECM. Basement membranes are sheet-like depositions of ECM on which various epithelial cells rest. Each type of connective tissue in animals has a type of ECM. For example, collagen fibers and bone mineral comprise the ECM of bone tissue, reticular fibers and ground substance comprise the ECM of loose connective tissue, and blood plasma is the ECM of blood. The plant ECM includes cell wall components like cellulose in addition to more complex signaling molecules. Some single-celled organisms adopt multicellular biofilms in which the cells are embedded in an ECM composed primarily of extracellular polymeric substances, that is EPS. Now let's talk of the molecular components of ECM. The molecular components of ECM are produced intercellularly by resident cells and secreted into the ECM via exocytosis. Once secreted, they then aggregate with the existing matrix. The ECM is composed of an interlocking mesh of fibrous proteins and glycosamine glycans, that is GAGs. Now let's talk the proteoglycans. Glycosamine glycans are carbohydrate polymers and are usually attached to extracellular matrix proteins to form proteoglycans. Hyaluronic acid is an notable exception. Proteoglycans have a net negative charge that attracts positively charged sodium ions, which attracts water molecules via osmosis, keeping the ECM and resident cells hydrated. Proteoglycans may also help to trap and store growth factors within the ECM. The different types of proteoglycans found within the extracellular matrix are mentioned below. Number first, heparin sulfate. Heparin sulfate is a linear polysaccharide found in all animal tissues. It occurs as a proteoglycan in which 2 or 3 hydrogen sulfide chains are attached in close proximity to cell surface or ECM proteins. It is in this form that hydrogen sulfide binds to a variety of protein ligands and regulates a wide variety of biological activities, including developmental processes, angiogenesis, blood coagulation, and tumor metastasis. In the extracellular matrix, especially basement membranes, the multi-domain proteins perlecan, argin, and collagen are the main proteins to which heparin sulfate is attached. Now, number 2, chondroitin sulfate. Chondroitin sulfates contribute to the tensile strength of cartilage, tendons, ligaments, and walls of the aorta. They have also been known to affect neuroplasticity. Number 3, keratin sulfate. Keratin sulfates have a variable sulfate content and, unlike many other GAGs, do not contain uronic acid. They are present in the cornea, cartilage, bones, and the horns of animals. Now the next part, that is non-proteoglycan polysaccharides. Number 1, hyaluronic acid. Hyaluronic acid is a polysaccharide consisting of alternating residues of D-glycuronic acid and N-acetyl glucosamine, and unlike other GAGs, is not found as proteoglycan. Hyaluronic acid in the extracellular space confers upon tissues the ability to resist compression by providing a counteracting turgor, that is swelling, pores by absorbing significant amounts of water. Now next is collagen. Collagens are the most abundant proteins in the ECM. In fact, collagen is the most abundant protein in the human body and accounts for 90% of bone matrix protein content. Collagens are present in the ECM as fibril proteins and give structural support to resident cells. Next is elastin. Elastins, in contrast to collagens, give elasticity to tissues, allowing them to stretch when needed and then return to their original state. This is useful in blood vessels, lungs, skin, and the ligamentum noci. Next is fibronectin. Fibronectins are glycoproteins that connect cells with the collagen fibers in the ECM, allowing cells to move through the ECM. Fibronectins bind collagen and cell surface integrins, causing a reorganization of the cell cytoskeleton to facilitate cell movement. Next is laminin. Laminins are proteins found in the basal laminae of virtually all animals. Rather than forming collagen-like fibers, laminins form networks of web-like structures that resist tensile forces in the basal lamina. Now, next, extracellular matrix in plants. Plant cells are ciliated to form tissues. The cell wall is the relatively rigid structure surrounding the plant cell. The cell wall provides lateral strength to resist osmotic turgor pressure, but it is flexible enough to allow cell growth when needed. It also serves as a medium for intercellular communication. The cell wall comprises multiple laminate layers of cellulose microfibrils embedded in a matrix of glycoproteins, including hemicellulose, pectin, and extensions. The components of the glycoprotein matrix help the cell walls of adjacent plant cells to bind to each other. The selective permeability of the cell wall is chiefly governed by pectins in the glycoprotein matrix. This was all about today's lecture. Hope you enjoyed and understood.